All right, recording begins for April 29th. This is ESM 303. Thank y'all for making it. This is Wednesday, right? Next week's week 15. So we've got today, next Monday, and then Wednesday, and then the semester's over. So that's 3 lectures left. And Wednesday's lecture, I've already promised I'll set aside a good portion of it for, Q&A. Come with your study guide, ready with questions about those topics, and we'll run through it all. We'll have no problem getting through the material on the final exam. Yeah. And then don't forget that next week, this week's your RES finals, right? Not RES finals, sorry, your lab finals. Next week, you're doing the RES symposium in lab, okay? So you need to have your files uploaded to Canvas before the lab section starts so that myself or your lab instructor can access those files for the projector. All right? It's also just it's got to be done. You're giving it, right? So get that PowerPoint done, get it uploaded before your lab section next week. Cool? Awesome. Very good stuff. Okay. Final paper's due at the end of next week, Friday, by 11:59 p.m. All right? You can turn it in before then if you want to. I'll grade it and get it, in the grade goal. Okay. Any questions about that? Yes?
Maybe you already did this, but not listening. Is the final going to be on Canvas so we can test different grades?
Am I going to post the final exam on?
Like the assignment, so when you put how many points is, you can put in like a test grade to see how it affects.
It should already be there.
It might be. It wasn't from last week, but
Oh, I can't project the grades because it'll, 1 second. Version. Exam. Oh, final exam is not Friday. Yeah, cool. I can fix that for you. Final exam study guide. The final exam is not Friday. Final exam is, boom, my class schedule. Just get 100%. Just do that. Why not, right? Okay, 1 second. Let's see when that exam is. It's Wednesday, that's right, good. Final exam Wednesday, let me just adjust this, and then it'll be available for you. Boom. Edit. Wednesday. Boom. It's at 10:20, correct. 10:20 a.m. Okay, let's adjust this. Edit. I know this is very exciting. Just bear with me, folks. Wednesday, 10:20. Save and publish. You saw me just edit that. I literally just did it. And now it's changed it back. Is it because I hit the back button? I don't think so. There it is. Beautiful. It's published. Boom. We'll move it into final exam. Publish this. Publish module. Edit. There. Now you can put your score in there. Yeah. All right. Any other questions? Final exam study guide's posted right here. Boom, boom, boom. Okay. Then let's go ahead and get back into the lectures. We'll wrap up the mimicry stuff. Let's confirm that this is working. All right, great. All right, so folks, we've been talking about interspecific interactions, okay? We talked about competition. We talked about predation, okay? And now we're talking about prey animals, okay, and their defenses. So we talked about apos. So we talked already about, hey, they can have physical defenses. They can have chemical defenses. We talked about all that sort of stuff previously. It's previous, don't worry about it. It's somewhere here. Remember we talked about it. Some things are poisonous, some things have defensive structures. Remember we talked about plants and thorns and things like that. All right, so there we go. Chemical defenses, cryptic coloration, flashing coloration. There's the slide. I don't know how I missed it. Also, sometimes when something's poisonous, it'll have what's called aposomatic coloration, where it warns predators that, hey, I'm poisonous, okay? And it's usually something that's very flashy so that it's easily noticeable and that the predator population can sort of adapt to identifying the pattern as this is lower quality food. Sort of makes sense? So it's, sometimes you wonder, like, what's the benefit of having all these flashy colors? And it's like, it's really easy for the predators to say, okay, yellow and black and flashy means that it's bad food. All right? Awesome. But if there's something else putting all the effort and the resources into generating poison inside of its own tissue, or these defensive structures like stingers and fangs and all this other stuff, then there's going to be an advantage to looking like that thing. You see what I mean? You're less likely to be consumed if you look like something that is poisonous. Just makes sense. So other species evolve to mimic things like bees or wasps, or in the case of the burrowing owl, it makes a rattlesnake hissing noise to deter possible predators. But this is mimicry, and it's not really reinforced, because if something goes up and eats one of these mantid flies, the mantid fly doesn't actually have the wasp's defenses. So there's not actually a negative repercussion for the predator to try to eat these, which means that that predator is now not learning, like, oh, that coloration's harmful. All right? So these species are sort of piggybacking on the species that actually have defenses and are actually warning predators, like, listen, don't bother with me. I've got defenses. You don't want to mess with me. All right? These species just look that way. And this is a special type of mimicry called Batesian mimicry, okay? Where a non-poisonous species looks poisonous, or a species without defenses mimics something with defenses. Reasonable enough? Cool. So that's Batesian mimicry, okay, as opposed to Mullerian mimicry, all right? Where in Mullerian mimicry, poisonous species look more like each other. So that means that they're all reinforcing. You've got all these different species that are all poisonous, and they all have yellow and black coloration. And so that reinforces predators, like, oh my gosh, everything that's got black and yellow stripes stings me. Everything with black and yellow stripes is bad to eat. I don't need anything with black and yellow stripes. So this sort of mimicry, a Mullerian mimicry, it reinforces the predator's avoidant behavior. Whereas Batesian mimicry sort of weakens the impact of having the aposomatic coloration. Are there questions about that? They're very important in how these things are reinforced and how effective the defenses are. Yeah?
What would be the difference in Mullerian mimicry and just like a shared trait because they're all the same genus?
So if they are all closely related, and so they have, that would be a shared characteristic because you're closely related. But in Mullerian mimicry, in true Mullerian mimicry, it's convergent evolution, where species are getting that coloration not due to common ancestry, but because there's a selective advantage for it. Does that make sense? That's the division. Yeah. Awesome. Additional questions about this? And like I said, it's got serious repercussions for how effective these different defenses are. There's lots of really cool papers about it. In my Principles of Ecology class that I haven't taught for a while, I've got a really good article about these invasive poisonous frogs and the snakes that are native there, and there's a lot of cool stuff. Anyways, why is this effective against predators? Because it reinforces, like, look, this coloration means I'm poisonous. Fair enough? All right. So additional defenses. Doesn't have to be chemical. It doesn't have to be a coloration. It doesn't have to be that I go, bah, and scare you like a damn grasshopper. We've talked about the grasshoppers and how I feel about them. You've also got behavioral defenses. So there's some classic examples out there, like meerkats. You know, that was a popular example back when I was a student. Okay, because that was when meerkats just took the world by storm, and everybody was like, oh, they're so interesting, and they're so cute, and did you know that they warn each other, and they have a complicated community? And it's like, yep, that's pretty cool. And every class mentioned it, and every class would play like Meerkat Manor clips, and it drove me crazy. But that's neither here nor there. Meerkats. They do alarm calls, okay? So these are behavioral defenses against predators. Remember when we were talking about survivorship curves, or not survivorship curves really, but oh my gosh, I'm blanking on the example now. Remember when we were talking about how some species do worse when they are at low abundance, all right? It's the, oh my gosh, the Ali effect. Thank you. Yes, very good. This is one of the key reasons that we get Ali effects, right? Is if there's some behavioral defense like living in packs or alarm calls where you do better when there's a bunch of you because you have more defenses, then that leads to the Ali effect, for example. All right, cool. So we got behavioral defenses like alarm calls and living in groups. And then I've already brought up predator satiation, okay? But we've talked about masting a little bit. You know about masting? It's in oaks mainly, right? You talk about these oaks. They coordinate producing fruits, where they all produce a bunch of acorns at the same time. And then they produce a few acorns in the other seasons. How does it happen? There were a lot of theories, but most evidence supports that it's just due to cycles of temperature. And there's like a molecular clock, where in the temperature cycles, hot to cold enough times, they all reproduce and generate acorns. But there's way more interesting theories out there, like, oh, they're all communicating via their mycelial networks and all this stuff. But the evidence doesn't really support that. So anyways, the point is, what's the benefit of masting? Why not just make all the seeds that you can every single semester? Why not just make as many acorns as you can every semester? You can tell this is going to be on recording. I don't know about you, but I am so ready, speaking of semesters, to get through this one. This is good. This is exciting stuff, though. Okay. I'm locking in. I'm looking at all of you. I'm remembering that you all are spending money for this, and you deserve for me to be engaged, and there's information for me to convey to you. Why, if you're an oak, don't you just make as many acorns? Is this better? Why don't you just make as many acorns as you can every single year? Well, if you're putting resources into reproduction, they're not going into vegetative growth. So just making acorns every 5th year, that means that 4 out of those years, you're able to focus on getting big and strong and making lots of leaves that have high photosynthetic rates. Life is good. Here's the other cool thing. If you're not making acorns, then the things that eat acorns don't have a food source, so they're not reproducing. So you keep the predator population lower, the population of things that eat you gets reduced because there aren't as many of you around. All right? And then, at a certain point, you just flood flood the ecosystem with individuals. You make a bunch of acorns, or all the cicadas emerge, and the predator population starts to grow because now there's a bunch of acorns present. We'll just keep using the acorn example because I'm a plant biologist, okay? The predator population begins to grow. The things that eat the acorns go whoop, 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 whoop. But then there's no acorns the next season, so the population crashes. And you can satiate that small predator population pretty quickly by making a ton of acorns, right? So satiation, you fill up all the predators. The predator population doesn't keep eating, all of your offspring. So that's another defense that you can have against predators, is just make so many of your offspring that they eat all they can, and there's still plenty of offspring left over. Cool? Love it. Oh, that's the last slide. Okay. Well, that's the parasitism, parasitoidism, herbivory, and true predation PowerPoint, which can now move us smoothly into the mutualism, commensalism, and amensalism PowerPoint. Because we're still talking about interspecific interactions, right? The idea is that we've moved up in the scale, and we're slowly moving towards community ecology. And really now we are talking about community ecology in that we are talking about interactions between different species, okay? So interactions between different populations. So we're talking community ecology. We're just introducing it as if it's always two species interacting. To jump to community ecology, you just go like, okay, now instead of 2 species, there's 30. And each one has its own different set of interactions. And so there's a different model for each one's interaction. And some are competing, and some are eating each other, and some are parasites, and some are hosts. But you just put all of these different models on top of each other, and you have a big population. Nothing too wild, all right? We understand it at the smaller scale, and then you just extrapolate it and say, now imagine there's 30 of them, Okay, so at this point, everybody's comfortable with what these positives, negatives, and nulls sort of mean. I'm assuming. Does anybody have any questions about those? It's all about fitness. In the interaction, does one individual get an increased fitness due to the presence of the other species? So if both species benefit, if both species do better, if species A does better because species B is present, and species B does better because species A is present, then it's a mutualistic relationship where both species experience a positive impact. And a positive impact is measured as increased fitness or an increased number of offspring generated, usually. All right? Cool. Because we're looking at all of this through the lens of natural selection and evolution. And if we do that, then all that matters is how many viable offspring you generate. How many copies of your genetics you pass on. Cool. Lynn Margulis. All right. So, we're going to talk about mutualistic symbiotic relationships. Symbiotic is not the same as mutualistic, and it's really easy to get them confused. Symbiotic just means that two things live in close proximity to one another, all right? They interact really directly. Sometimes one thing lives inside of another, all right? So it's symbiotic, like living together, okay? So a symbiotic relationship isn't necessarily a mutualistic relationship. A parasitic relationship can also be symbiotic. So people misuse those terms all the time, and now you can go out there and use them correctly, all right? And you can use that newfound power to correct people, which everybody loves. Whenever somebody says symbiotic, go, I think you mean mutualistic. And, that's okay that you're wrong, but you should really, read something probably. That's a great way to make friends and influence people, all right? So, symbiotic relationships just means that you live in close proximity, interact really directly, okay? Mutualistic relationship means both organisms do better with the other one there. So this is an opportunity for us to talk about the symbiotic relation, about the endosymbiont theory, okay, which is basically what explains the presence of organelles in cells, okay? Lynn Margulis has unfortunately passed at this point, but, whoa. All right. But she proposed this sort of theory, that there was a free living organism, and that free living organism tried to eat, it was a single-celled organism, and it tried to eat something that could carry out cellular respiration really effectively. But it didn't digest it. For some reason, that thing continued to live on inside of that host organism, all right? Which was the thing that became the host organism. And it was beneficial to the host. That, oh, now this thing that's inside of me is carrying out a ton of cellular respiration, making a ton of ATP, reproducing. All I have to do is keep getting sugars, you know? And if I just keep breaking down sugars inside of my cytoplasm, that thing will take care of the rest and just make so much excess ATP that all of my endergonic reactions can fire off. Like, sick, you know? It's a great situation. It's easier to understand from the perspective of chloroplasts, because with that, you're like, okay, I'm an organism that's floating around, I'm a single cell, and I can't do photosynthesis. And there's another smaller organism that carries out photosynthesis and makes sugars. So I go try to eat it, because I'm just a single celled organism, I'm just trying to eat, you know. I eat that thing, and then I can't dissolve it. And now it's inside of me. And now it's carrying out photosynthesis and pumping out sugars. And I'm breaking those sugars down, and I have this other free-living thing inside of me that is taking those sugar breakdown products and churning out a ton of ATP. So that endosymbiotic relationship, or endomutualistic relationship, allows additional complexity because you have additional energy production. This is the endosymbiont theory, and now it's widely accepted as where organelles came from, primarily the mitochondria and the chloroplasts. There's an additional first step here where you have to protect your nuclear genome first. So the first step up here is that the cell membrane for the single-celled organism starts to fold in for the prokaryote, and it makes another membrane around the nuclear genome, the main genome, okay? That means that these things aren't going to screw with your DNA by being in there. You've got your DNA all protected. And there's evidence of organisms now, prokaryotic organisms now, that are doing this. They are developing nuclear envelopes. Because this process it's still advantageous to have your DNA kept in a separate little sac. It protects it, right? So this process doesn't really stop, you know, like evolution doesn't work that way where it's like, well, that thing's gonna evolve, so I'm gonna stop. They're taking care of having nuclei, so we're not gonna do that, you know. So some single-celled organisms are still following this path, and there's now evidence, just like 2 years ago, of nitrogen-fixing endosymbiotic relationships, which is nuts, you know, because we've always learned that nitrogen fixation can only be carried out by bacteria, okay? And anything else that does nitrogen fixation has to have a symbiotic relationship with bacteria. But now there are eukaryotes that are consuming these nitrogen-fixing single-celled organisms and not killing them inside of their cytoplasm. And they're still able to do some nitrogen fixation. So new organelles coming soon. We'll see. Yeah, this stuff's super cool. And it's a great example of a mutualistic relationship where the mitochondria get a benefit, And the organisms, the cyanobacteria and the proteobacteria get a benefit of living inside of here. But now, at this point, there's been so much exchange of genetic material into the nuclear genome that mitochondria and chloroplasts can no longer live together. So this is a clear example of what we would call an obligate mutualistic relationship, where one thing can't even live without the other anymore. Like it's become so necessary that it's an obligate mutualism. It's not just like advantageous to have mitochondria, you have to have them. And then on a more like goofy, silly note, I always bring this up in my Bio 105 class. There's a series of horror novels and that were converted into video games that are all about what would happen if the mitochondria inside of your cells became self-aware and wanted to wanted to be in charge. It's called Parasite Eve. Check it out. There's cool stuff in there of you get this evil villain and, she's like controlling everybody's mitochondria. So she comes into the room and she's like, bah! And she makes all of your mitochondria just respire as rapidly as they possibly can. And so you burst into flames. It's pretty fun. It teaches you lessons about energetics and what the mitochondria are, It misrepresents the science like crazy. They're like, they've got their own genome and they reproduce on their own. And it's like, not really. They do have their own genome and they reproduce independently of the host cell, but it's reliant on the nuclear genome as well. Anyways, that's enough stuff about the endosymbiont theory, but I hope you enjoyed the discussion. How's this energy level working for you? Is this better? This is good? Yeah, hey, I'm liking it too, Okay, let's keep on keeping on. Get all that? That was just a joke. We're just having fun today. Okay, so mutualisms. I've already talked to you about obligate mutualisms, okay? They have to be there. You have to have the mutualistic partner or you can't survive. Now, this can be tricky because sometimes you can keep one member of the obligate mutualistic relationship alive in a lab setting, but that's not really the same, right? Like, okay, sure, you've got some species, certain lichens, all right, are fully obligate, where the fungus won't survive if it doesn't have the algal or cyanobacterial symbiotic mutualistic organism present. But you could keep the fungus alive in a lab setting, but it won't survive in nature. So you say it's obligate, even though technically we could create a condition where we could keep we could keep the separate members alive separately. Does that sort of make sense? All right, awesome. Then there are facultative mutualistic relationships, where if I'm a plant and I've got a mutualistic relationship with a pollinator, I provide it with an energy source, it provides me, it vectors pollen for me directly to other members of the same species. Cool, we both get a benefit, we love it. All right, I said I provided nectar, right? And I said that? I think I did. Okay, great. However, if my pollinator isn't there, I still survive as a plant, right? Maybe I'm carrying out asexual reproduction as well. Maybe I make stolons and I'm spreading that way as well as making flowers. Or maybe we've got sort of a dispersed mutualistic relationship going on where I have a bunch of different pollinators. And some are, more effective than others, but I'm also being pollinated by flies, all right, and ants. So just because I lose the bee that's pollinating me, that's like my highest efficiency pollinator, I might not actually perish from the landscape. So that's more of a facultative mutualistic relationship, where I'm not quite as dependent. So we can characterize mutualisms by hey, what's the benefit that I get? Do I get protection? Do I get an energetic resource? Do I get access to a habitat that I otherwise wouldn't get access to? You know, there's all these different things that could happen. And then we've got how dependent are the species on one another. All right. Then we can characterize them with how specific the mutualistic partners are. So flies, I've already talked about them, okay? They're generalist pollinators. Flies will pollinate like anything, and they're not really good at it. They don't have great efficiency. It's better if you're a plant to have a pollinator that is specialized to you, because that means that it's only going to be going between members of your species. So it's only vectoring pollen between organisms that are actually reproductively compatible. Flies go to any plant and they're just like, yeah, sure, cool, I think there's some resources here, I'll just land on there, and yeah, I'll suck out some of this nectar, great, I'm all over the place, I'm disgusting, okay? That means that they might never encounter two flowers of the same species, or by the time they do, they've lost the pollen, all right? So it's a generalist instead of being a specialist. You compare this to orchids, right, which have very specific mutualistic relationships with their pollinators, where they make their flowers look like individuals of that species of their pollinator. All right, to try to get them. It's, you know, this is oversimplified, of course, because it's not an orchid class, and I'm not an orchid specialist. But many orchids will be like, I'm going to look like a female wasp, and that's going to mean that the male wasp will come by and try to mate with my flower, and then I'm going to deliver the pollen, and it's going to go and find another orchid that looks just like me, and we're the same species, and it's going to try to mate with that flower too. There you go. You've got a very specific sort of mutualistic relationship there. Now, if the plant doesn't provide some actual benefit, then this could actually reduce the fitness of the pollinator. Imagine a situation where the orchid doesn't provide any nectar or anything. All it does is mimics the female of the species. Well, now that wasp is wasting energy trying to mate with these flowers. It's not passing on the genes. The population is going to take a negative, going to decline, and now all of a sudden it's a parasitic relationship. See how easy it is to shift between these? Things can look superficially the same, have the same sort of structures underlying the process, but if the end result is a reduction in fitness, then it's a different type of interspecific relationship. All right. Great. The duration of the intimacy. It's an interesting bullet point right there. So, you know, I've already talked to you about obligatory. If you've got, your mitochondria are never going to get out and live freely. You know what I mean? In many of the lichen symbiotic relationships, the algae or the cyanobacterium, it's not going to be free living. The fungus isn't going to be free living. They stay living together and they produce propagules that have both cell types together, and then they live together again. You know, it's obligatory. There's not a free living phase. Okay? Question, comment. It's slightly different because you can have this. It's kind of silly to have it as two different bullet points, honestly, now that I'm looking at it right here. It's slightly different because you can have an obligatory relationship, just implies that they both must, that if one partner's not there, the other one dies, right? That the species can't persist in the environment without the other one. But you could have a pollinator, let's say you have a very specific pollinator, all right? And the population is of an annual plant that completes its life cycle each season and doesn't asexually reproduce at all. Well, now what you have is an obligate mutualistic relationship with that pollinator, where if that pollinator dies, then you die, right? Because you can't produce seeds for the next season. But the pollinator is still free living. The duration of the intimacy is short. It could just be like, The interaction only occurs during when I have my flowers out. All right? And then for the remainder of the season, I have no interaction with that pollinator species. Do you see how there could be situations where, okay, awesome. I don't have a real world plant example to throw out at you, but that's exactly what would lead to an obligatory relationship where the individual is still free living. Yeah. Awesome. Okay, so we have to talk about mycorrhizal associations. At Humboldt State, at Cal Poly Humboldt, all right, you can see, my age is showing, okay? People tend to have a general interest, or you encounter people with interests in mycology here, way more than at other institutions. I used to teach at Riverside City College, all right? And I came in to talk to them about mutualisms, and we were talking about lichens, and I was just like, okay, so everybody knows about lichens, blah, 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 blah. And they were like, what's a lichen? I'm like, oh, wow. So you know that yellow, crusty stuff when you were walking in on that rock? And they're like, yeah, what is that? And I'm like, that's a lichen. Here, that sounds ridiculous. Like I would assume that 95% of this class is familiar with the fact that a lichen's a symbiotic relationship between a fungus and an algae or a cyanobacterium. And you probably also in your head, many of you are also like, well, there's other species that can be the photosynthetic component of there. And it's like, yeah, very good, awesome. You're probably also familiar with mycorrhizal associations. And that's not the case everywhere. But here it's taught more aggressively. We've got mycologists on staff. We have a mycologist on staff on campus. That's not a thing that most universities have. So that information kind of just makes its way through the student body. So while all of that to say that, well, this probably feels very basic for most of you and like something that you've encountered before, not everybody in the room's in the same boat. So we're going to talk about mycorrhizal associations. Okay, I brought it up once before, I believe, when we talked about accessing resources in soils. Okay, and how certain resources are more easily accessed by expanding your root network with a mycorrhizal associate. What's mycorrhizal association? Okay, fungus grows around, maybe into the roots of a plant, extends its hyphae, all right? The hyphal filaments are thinner than roots, so they can get through spaces that the roots can't get to, and the hyphal network gets bigger, right? The mycelial network explores more area of the soil than just the roots. There are two main categories, two major groups for mycorrhizal associations: ectomycorrhizal associations and endomycorrhizal associations. Okay? Ecto means outside, all right? Endo means inside. Those are classic prefaces, and you'll see them with all sorts of in all sorts of words. If you're not familiar with them yet, you'll start to see them all over the place. So the ectomycorrhizal associations, the mycorrhizal, the fungus still grows into the tissue, but it doesn't actually penetrate the plant cells. So while it can grow, it forms like these sheaths around roots, and it can grow sometimes between the different cells. It doesn't actually grow into the plant cells. Whereas in the endomycorrhizal associations, okay, you grow into the plant cell. Arbuscular mycorrhizae, all right, form these structures called arbuscules. An arbuscule, it has the same word root as like Arbor Day or an arboretum. It's called an arboretum, right? Not an arboretum. Doesn't that sound like, is anybody with me on this? It doesn't sound right. Arboretum, 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 arboreum, arborea, arboritor. You're not giving me anything. Nobody's got nothing, not helping me. It's Arbor Day. Arbor Day, Arbor Day, Tree Day. You're supposed to go plant a tree on Arbor Day. Everybody knows Arbor Day. You all celebrate it, right? That's great. Thank you very much. Okay. An arbuscule mycorrhizae, they form these arbuscules. Cool? Whatever. Get off my case. So we've also got ericoid mycorrhizal associations, very important in this area. Ericoid, ericaceae. Remember the ericaceae, that family? It's got like vaccinium in it. The plants tend to have those urceolate flowers. Look, we're connecting lab and lecture material, all right? You'll notice that if you go into the redwood forest, where the dominant species is Sequoia sempervirens, much of the understory is made up of members of the ericaceae. The ericaceae has a specific type of mycorrhizal association. There's a specific species or specific group, taxon, of fungus, of mycorrhizae, fungal mycorrhizae. Sorry, I'm engaging again. There's a specific taxon of fungus, all right, that forms these ericoid mycorrhizal associations. One of the reasons that the understory of the redwood forest is so abundant with members of ericaceae, you've seen multiple, not only the vacciniums, right, but also the salal, Gaultheria, thank you, Gaultheria chalone, all right, but also that species, okay, and there's others. It's because the duff of the redwoods inhibits many mycorrhizal associations, but it doesn't inhibit the ericoid mycorrhizae, okay, which allows those members of ericaceae to succeed in the understory and indicates to you how important this mutualistic relationship is. Even though it may not be obligate, the species could survive on its own without its mycorrhizal symbiont. It allows species that have them to succeed over species that don't. All right. Next one, orchidaceous mycorrhizae. So I already brought up orchids as examples of, very specific pollinator and pollinatee relationships. Okay. There's also a very specific type of mycorrhizal association with the orchids, okay, where you need to have. You ever see an orchid seed? Yes. How big are those orchid seeds? Small, that's right. Hey, if I'm giving, you see how much energy I'm putting out? If I'm giving it out, I need it back. How big's an orchid seed? Small. You're darn right they're small. Some of the smallest seeds out there. They look like dust. All right. Hey, we do orchid culturing for the greenhouse. Go check out the greenhouse. Talk to the greenhouse manager. Ask if you could do some tissue culture of orchids. Why do we have to tissue culture orchids, you might say? Why can't we just take the seeds and plant them in the ground? Well, small seed, very few resources, okay? So many orchids have a parasitic sort of relationship with mycorrhizae, which is wild, all right? So now I'm a little orchid seed, and I've got no resources. So I'm there in the soil, and I put out these signals, and I'm like, hey, I could use a mycorrhizal associate over here. Like, anybody want to be an endomycorrhizal associate with me? You can grow into my cells. You can make an arbuscule, huh? And then so they go, okay, great, I'll grow into you. And then the fungus grows in, and I go, I got you, you chump. And I go, chunk, and I change my structure a little bit, and I chop off that mycorrhizae inside of me. Then I break it down. I get the energy from that, and now I'm good. And now I'm an orchid. And I'm like, now I'm just going to go trick some wasp into trying to have sex with my flowers, And that's why orchids are deceptive plants, and, they're also bad. They're bad plants. I don't know. Like grasshoppers. It all comes back together. There's a lot of animals and plants that I don't like, and that's the point of this lecture. That'll be on the exam. Which one of the following organisms is John mad at? The answer is all of them. It's like just be a phone, am I right? Flower? Cool. There's other complex symbiotic relationships between orchids and their mycorrhizae that you can totally look into, but I think that's the coolest one, where the orchid becomes parasitic on the mycorrhizal associate. And then, that wouldn't really work unless sometimes orchids didn't do that, and sometimes they don't. But orchids have got to have some other source of energy because they've got no nutrient reserves in those little dusty seeds. Whoa! I don't know why I have those arrows coming in. Okay, what sort of benefits can a mycorrhizal association give its partner? You can get nutrients that you couldn't otherwise access. You can get more water. If you got a big sheath of fungus around your roots, that can actually protect the root tissue from infection, all right, by pathogens. The things that could eat the roots of the plant can't necessarily eat the fungus. That's also beneficial, all right? Pretty cool stuff. There's also like this last bullet point, which is pretty cool. We haven't really talked a lot about nitrogen fixation in plants as a symbiotic relationship. Do I have it coming up? Not really. Not really. That's disappointing. I should have more about this. That's okay. That's okay. We'll talk about it a little bit right now. Nitrogen fixation. We talked about it maybe with the in the soils lecture. That's why I don't have it here, because we talked about it in the soils talk. Remember, there's all this nitrogen in the atmosphere. There's so much nitrogen. We're breathing it in right now. You're surrounded by nitrogen gas, all right? And you need nitrogen. You need it for your amino acids, and you need it for your DNA, for your nucleotides, all right? Nitrogen's important. But we have to eat things that have nitrogen in them already, because we can't take that nitrogen and fix it. Not yet, not until that new organelle hits the market, all right? Get a copyright on that son of a gun, inject that into everybody. No more need to eat proteins, all right? Because now you make your own amino acids. That's not how it works. We're near the end of the lecture, so I'll just give you something fun. I'll give you fun facts. That's like a good way. That's the thing you can take with you. Nitrogen, all right? You know it. You love it. We'll talk about how mycorrhizal associations are associated with nodulation rates of rhizobia in next Monday's lecture. But you know the flavor profile of umami, all right? So that flavor profile, it gets like a new sense, a new flavor profile that wasn't understood until, relatively recently. The person who discovered umami actually developed MSG. Isn't that interesting? That's pretty cool. Also, why do you have that? And why does your body go like, ooh, yeah, cool, like meaty, like savory? It's because your body is detecting the presence of polypeptides, of proteins, of amino acids. And so just like how sweetness gets your body to eat more sugar, all right, and saltiness gets your body to get more ions, all right, which you need to have, umami gets your body to eat more proteins, which are sources of nitrogen, all right? You got to have nitrogen, and you can't fix it yourself, so your body's developed a way to tell you, like, hey, that has nitrogen in it, eat more of it. Isn't that good? There you go. There's your little tidbit of information. I'm done. All right? I hope you all had a great time in lecture today. I certainly did. And I'll see you again on Monday for our last full lecture of ESM 303. By Wednesday, have your study guide started, okay? So you can come in with questions. Cool? Thank you. Thank you. Thank all of you. No, stop applauding, please. No, it's not necessary. No, I appreciate it. Oh, thank you. Thank you so much. No. Thank you for watching this recording.